Hello and welcome to Coast and Country, powered by the science of the Connecticut Agricultural Experiment Station. Coming up in this special edition, all about Plant Science Day 2025 on the station's 150th birthday, we talk with Sarah Nason. Talks about the station's research into PFAS, the forever chemicals that affect all of us and are everywhere. Nate Westrick on new emerging strawberry diseases affecting our farms. Phil Armstrong updates us on the station's mosquito monitoring program and West Nile virus. Gudaz Malai talks about the latest research on ticks and new tick species coming to Connecticut. And Lindsay Triplett talks more about the 150 years of the Connecticut Agricultural Experiment Station. But first, we catch up with Dr. Jason White, director of the station, and his opening remarks for Plant Science Day 2025.
This is my 27th Plant Science Day as a station employee, 5th as director. Last year was the only time I ever remembered it raining, so at least we have beautiful weather this year. So just a few updates on things at the experiment station. This is just today's schedule, which you all have. And as usual, you'll be hearing short talks from 3 of our scientists on 3 different topics that we think will be interested to you. We have Scott Chasky, who is going to be our Johnson lecturer, which I think also will be a very enjoyable topic. And then Century Farm Award and some other speakers to kind of close things out for the day. In terms of our barn exhibits, which are in these barns right behind us, we have our 5 different departments. Each has a barn exhibit. But first we have a kind of general barn exhibit, because I'll mention this in a minute, but we're celebrating our 150th anniversary this year. There's a barn exhibit there that kind of captures some of the science that's been done over the last 150 years. But our Valley Lab, there's a presentation on do-it-yourself deer repellents, which is really interesting. Plant pathology and ecology, which you'll hear about as a short talk as well, but some of our work on managing apple diseases, environmental science and forestry, using organic and inorganic amendments to improve soil for growth. Analytical chemistry, you can learn about some of the work that we're doing on using nanoscale bioemulsions to protect tomatoes and other fruits after harvest. And then entomology has a presentation on the invasive tiger mosquito and how that increases risk of heartworm to dogs. So in addition to that, back here on the other side, we have 33 outside exhibitors for you to view. So just as kind of the station update, I have to put a report together for the governor each year, so we pulled this information out of it. You can see kind of our funding breakdown. The total agency funding is about $15.3 million for the year that just closed. I always say you'll never find another agency that does so much with so little. About $8.7 million of that's the state general fund, but the rest is other sources, including federal funds. Obviously, you'll see all of our research today, but it falls into these 4 major core areas: agriculture, the environment, food safety, and public health, plus the 5th area being all the outreach and extension type of projects that we do. So I usually ask 2 of our programs to provide some updates, and so this year I went for the tick testing laboratory and also the mosquito program on the next page. So I'm not going to read through this because there's a lot of updates, but I think most of you know that our tick testing laboratory has been getting increasingly large numbers of submissions, and we're finding lots of different species carrying lots of different pathogens. So this is just kind of summarizing that, and as a research organization, in addition to putting our findings out for the public, we also put them in scientific journals so our peers can learn what we're doing, and that kind of summarizes some of this work. Mosquito program, as some of you may have heard this week, it's a very active season for West Nile virus, so I encourage you to visit our both the barn exhibit and the plot exhibits on that topic. But as I think we're all aware, we put 108 sites, trapping sites around the state. basically trying to surveil the viruses that are in the mosquito population, because that's a leading indicator of what could show up in the human population. So again, focusing on some of the research and outreach that we're doing in that program. Our two major construction projects, the first one is one I have talked about a couple of years now. This is our Valley Laboratory renovation. For those that aren't aware, our main location is in New Haven, but we also have a laboratory in the Valley Lab, a laboratory in South Windsor called the Valley Lab. The building, which you can kind of see on the left, was constructed in 1940 and has not really been renovated since. So we have a $36 million renovation of that structure and then addition of state-of-the-art laboratory space behind it, which is the rendering you see on the right. So that project went to the bid room, or is in the process of going to the bid room and then out onto the street for bids. So we are hopeful to have groundbreaking later this fall on that project, which will be quite the undertaking because this started quite a few years ago. The second construction project is done. So this was the greenhouse project in New Haven. This was a $1.6 million greenhouse project. We actually have 5 greenhouses down in New Haven, but they all date to the 1950s or so. To enable our scientists to conduct the type of research that they need to conduct, we needed updated facilities. So that project was completed. We had a ribbon cutting May 22nd, and I've just got some photos, including some of our scientists now using this resource. So as I mentioned, this is our 150th anniversary. So the Connecticut Agricultural Experiment Station was founded at the end of July in 1875. We were established as the first agricultural experiment station in the country. The story behind that is Samuel Johnson, who was a Yale chemistry professor at the time, spent some time in Europe, saw the agricultural experiment station system that existed there, which was where you had technically trained scientists whose job it was to work directly with growers. Thought that was a great idea, came back and petitioned the Connecticut State Legislature for the first ag experiment station, which was established, as I said, 1875, a 2-year budget of $2,500, 2 employees, and that system has since grown. The U.S. Agricultural Experiment Station system now, there are ag experiment stations in every state in the nation, plus a couple of other countries and territories. So we're looking at 13,000 scientists now working in this system.
So what are PFAS? They're a group of chemicals, and from a chemistry definition standpoint, they're a group of molecules that all contain carbon-fluorine bonds. So that's a definition that includes a lot of things, because all of those molecules can also include anything else, and it means that there's really a lot of PFAS. There's over 10,000 of them listed on the US EPA website. And the thing about carbon-fluorine bonds is that they're very strong. So PFAS don't degrade over time. So it's earned them the nickname forever chemicals. They've been in use since the 1940s, and they are extremely useful molecules. They can be used to make up all sorts of different things. They're used in firefighting foams and food packaging and waterproof coatings, stain-proof coatings, but their widespread use has also led to them being widespread environmental contaminants. And just to emphasize, these are really widespread, very useful chemicals. The firefighting foams have led to them being used a lot and spread all over the environment. They are used in things that are in contact with food, things like pizza boxes, fast food packaging, anything that makes paper not act like paper as much, so it will repel grease or water. Waterproof gear like Gore-Tex boots or coats, and it's led to it being found frequently in drinking water and soils. And that ends up being a problem because PFAS are actually quite toxic to people. They have a wide range of potential health effects, including risks of thyroid disease. They can impact blood cholesterol levels. They can decrease the body's response to vaccines. There's effects on fertility and on the development of unborn children. So it's really a problem. And as a chemist, one of the things we look at for contaminants is how much of a contaminant it takes to be toxic. And for PFAS, that number is very, very small. Even very, very tiny amounts of PFAS can potentially cause health effects. Because PFAS are so ubiquitous, they're actually already present in the blood of virtually all Americans, and not just Americans. This is true worldwide, but the data I have to show you today is from the NIH, which surveyed people in America. So the good news about what we see in this data is that some PFAS concentrations are decreasing over time. As awareness has been raised that PFAS are a problem, some of the most commonly used PFAS that used to be in many products have been banned globally, and their use has been decreasing, and we can see corresponding decreases in blood concentrations. So the bars on the left, those are from concentrations measured in people from 1999 to 2000, and then the bars on the right, which are much lower, are from 2017 to 2018, and that's the most recent NIH survey data we have. It's really showing a decrease. It's showing that the policies are having an impact. This is something that likely has a real impact on health, but it's something that's hard to measure. It's something that many people are being exposed to, and it's something to be concerned about because it's so ubiquitous that we can see it in virtually everybody. And while it's good news that some of these are decreasing, the not so good news is that the replacement chemicals that are now being used instead of these are also PFAS in many cases, and so those may be having a corresponding increase. Hopefully the replacement ones aren't quite as toxic, but there's not as much research about those, so it's a never-ending cycle of figuring out what we can use to cover all of those uses that PFAS have, but also not be causing toxicity to a wide portion of the population. If you follow the news, you've probably heard about PFAS quite frequently. These are some headlines that I looked up. These are just things from the past couple of weeks or so, and they've been talked about in a wide range of news outlets. And it's not just your crunchy environmental news sources that cover this stuff. We're talking about Fox channels, business journals, military websites, are all talking about how PFAS are having links to health problems, how they're being in drinking water, how there's lawsuits. So they're really a big topic at the moment. Specifically in Connecticut, these are some headlines that have been in the news in the past year. There's been news about Connecticut banning PFAS in certain products. Connecticut's really been a leader in the country on PFAS elimination, sort of stopping contamination from the source, looking at bans in cookware and firefighting foams and firefighting gear and paper products. There's also been talk about our new program with PFAS in farm soils. Because PFAS are in so many consumer products, they end up in wastewater. It goes through the sewers, the water goes to a wastewater treatment plant, and everything put out by the wastewater treatment plant ends up having PFAS in it. And this includes biosolids, which are also a great source of nutrients for plants and have been used as fertilizers. This practice isn't as common in Connecticut as it has been historically in other places, but it is definitely still a concern. For some background information, we find PFAS in virtually all soils, and it's possible that they get taken up by plants. With maple sap, if the tree takes up any contaminants and they end up in the sap, in the syrup production, the sap gets boiled down so much that contaminants could potentially be really concentrated. So it's a food safety factor to be able to look for contaminants that are in the sap to make sure that syrup would be safe. In our lab, functionally, we've used this project as a practice, as like a warm-up run for doing our soil on actual farms. So this graph here shows our detection rate for finding PFAS in the New England soils that are part of this project. And again, this is something that I would expect to be fairly similar in farm soils. And overall, we find PFAS in every single sample. We found at least two individual chemicals in every single sample we've tested out of the 42 that have been included in this project so far. PFOS, which is one of the common historic PFAS that was used, we found in every single sample. PFOA, PFDA, PFUNA, those are some of the common PFAS we're seeing too. But again, in this project and in other projects I've worked on, we have never analyzed a truly blank soil. Pretty much everything has some amount of PFAS that we can see. In terms of ongoing research efforts, we do have projects on PFAS remediation, ways of removing PFAS from soil. We've done some work with biochar. We have some active work on PFAS phytoremediation, so using plants that can grow and draw the PFAS up from the soil. We have some investigations on how microplastics might affect PFAS accumulation in different organisms. I have a new project working with someone in New Jersey. We're getting some like fish and plankton and seawater samples to start looking at PFAS in marine field chains. And then the controversial, canceled and then reinstated grant that I have is about bioaccumulation of PFAS in agriculture and in culturally important food webs in northern Maine. So that's in direct collaboration with farms and tribes in northern Maine looking at products and regions that they are concerned about. So it's research that's really making a difference to people. It's research that's enabling us to improve our methods to the extent that we can regulate PFAS in a better way.
So strawberries are, while not the highest acreage crop in Connecticut, by far the most valuable per acre. And that basically puts them into this kind of important niche for the state. First of all, agritourism is huge in Connecticut, but also we have hundreds and hundreds of small farms all over the state, and strawberries are the earliest producing fruit. And so it is basically this economic anchor that will keep these farms productive through the spring season until they can get other things like squash and tomatoes and such that they can sell to make it through the rest of the growing season. The issue with strawberries is that for a long time, because Connecticut and the Northeast in general has not been one of these really, really high density strawberry production areas, it actually hasn't been that difficult to grow a profitable crop. We haven't had as many challenges as other parts of the country like California and Florida. But in the last few years, we have, for one thing, climate change. We have warmer winters. Some of these pathogens that used to be able to survive down the south but not up here are managing to adapt to our climates. But also there's just more plant movement. I have individual farms that I work with that will order plants from North Carolina and Indiana and Canada and California all at the same time, which basically means that if a pathogen exists anywhere in the country that can cause damage to Connecticut farms, it will eventually make its way here. And we have a different growing system than a lot of the rest of the country, and so the issue is that if something works for controlling this pathogen in Florida, it doesn't necessarily work here. We have to do research to basically rewrite the book. Even if you view some of these small farms around the state as not being large strawberry operations, without strawberries, I would say a few hundred small farms in Connecticut and the Massachusetts area just wouldn't be economically viable. It is actually, you can get about $50,000 to $70,000 an acre for a U-pick strawberry operation, which blows most of the rest of these crops out of the water. So without this, a lot of these small farms that we depend on for local produce in our area just don't exist anymore. Some of the research that we're focused on is actually just by far the gold standard for dealing with plant pathogens is genetic resistance. We want varieties of strawberries that won't die when they interact with these pathogens. And these varieties exist, but actually there is not any guidance anywhere in the country for it, because in the parts of the country where these pathogens came from, mostly the southeastern US, they just grow different varieties. So you can imagine something called Florida Brilliance probably doesn't grow that well in Connecticut. So we've had to do large-scale varietal trials for the last couple of years to basically, where we grow out these plants in fields in Connecticut in different regions, and then we just spray as much of these pathogens as we possibly can on them and see which ones survive well. And we already have guidance that we've been spreading out to the farmers. So that's one thing, but also many of these pathogens, while we're calling them pathogens, they can be very destructive. Many of these will actually associate in sort of a relatively asymptomatic way that you might not even notice under certain conditions. We know that they can transition from what we call an endophyte, which is an asymptomatic kind of fungus that will colonize plant tissue but not do much damage, over to a pathogen. And we're currently working to identify those triggers, things like drought stress, things like plants not being fertilized correctly, things like other pathogens kind of biting away at the roots. And so we think that if we can get all of the different factors that are involved in that transition, then we can basically keep these plants alive whether they get infected or not. I get a lot of calls, and I do probably 2 to 3 farm visits a week to go out and try to make sure that we understand the issues. And they are very different. Sometimes someone will describe a field over the phone to me, and I'll think, okay, I know exactly what's going on. You show up and you realize, like, no, they have slightly heavier soil than everybody else. They have a different variety, and we'll realize that it's actually the opposite of what we thought. And so it requires an incredibly local approach. For most of the Northeast, actually, there's been this idea that some of these big institutions like Cornell will put out guidance, and as long as they're doing that work, that it works for everyone. And we've been finding over and over in the last few years, not just in strawberries, but in nearly all of our crops, it's just not true. We have different farms, we have different soil, we have different problems. When people think about climate change being a driver, they just assume warming winters are creating the problem, and that is probably part of it, is that some of these southern pathogens haven't historically been able to survive in cold Connecticut winters. But that I actually don't think is the biggest issue. It is these basically drivers of extreme weather. The thing that has been causing a problem, not just in strawberries, but in nearly all the crops in the state, is that we'll end up with a 3-week period where it just doesn't stop raining, and warm weather with lots of water is just, fungi love them. The fungal pathogens love it. And so if you get the root system that just doesn't dry out for weeks, and then suddenly you have a 3-week drought right after it, there's almost nothing you can do to prepare for that. And so I'm not going to paint a picture as if we can't do anything in the face of climate change. Even within our varietal trials, we're trying to find ways that we can grow these plants that are just by like hilling them up maybe, we can give more room for the roots to dry out in between these rains. And so we can tweak the agricultural practices. We have the ability to adapt, but it is very different. And the challenges are coming faster than we'd like.
Yeah, so our group is monitoring the risk of mosquito-borne viruses that can cause human disease. And the way we do that is by trapping and testing mosquitoes at locations throughout the state. So we have 108 trapping stations in locations all over the state where we collect the mosquitoes, bring them back to our labs in New Haven. All the mosquitoes are identified to the species level and then tested for virus infection the next day. And the reason for this is we're really trying to assess the risks for these diseases, and really looking at infection in the mosquitoes gives us advance warning for the risk of human disease. So this program has actually been in place since 1997. It was originally set up to monitor for the risk for Eastern equine encephalitis virus. And then in 1999, the first big change was West Nile virus was introduced into North America, and has now become the dominant mosquito-borne disease in the country. And we see this virus every year. And so we increased our trapping presence throughout the state. And over time, we are seeing changes in the diversity of mosquitoes and the number of mosquitoes over that 25-year period, where mosquitoes are becoming more diverse. We're not losing species, but we're gaining new species. Some of these are coming from East Asia. We have the Asian tiger mosquito and Aedes japonicus, which are invasive species that have come in recently and are becoming established here, as well as we're seeing species that are typically found in the southern US move northward into our area. So mosquitoes are becoming more diverse. They're also becoming more numerous. Our seasons are lasting longer because with warmer weather and with a warming climate, the climate is more hospitable. So West Nile virus is something that we see every year in Connecticut. The virus is maintained in a bird-mosquito transmission cycle. And humans will acquire the infection when the mosquito picks up the virus from a bird and then feeds on a human. We encourage people to take matters into their own hands, and personal protection measures are the first line of defense against these diseases like West Nile virus and Triple E virus. There are no vaccines for use in people. There are no treatments. So what you can do is reduce your exposure to mosquitoes, and that involves things like wearing a mosquito repellent when outside, particularly between dusk and dawn when mosquitoes are most active. The mosquitoes that transmit West Nile virus are most active in those time periods, as well as just covering up, wearing long sleeve pants and shirts when outdoors when mosquitoes are biting. Those are probably the two biggest things you can do, protect you and your family. Also around the home, just look for any pools of standing water that could collect in wading pools or in bird baths or pots, whatever, wherever you see, or clogged rain gutters, any standing water can serve as a source of mosquito breeding. So you want to eliminate those sources of standing water as well. So there are 50 different mosquitoes in Connecticut, and the mosquitoes they're all very different from one another. They specialize in certain habitats. So for example, the mosquito that transmit West Nile virus, it tends to be found in more built environments. The larvae will breed in standing water around people's homes or in those storm drains that you see on the side of the road. If there's not enough rainfall to flush them out, they'll breed in those storm drains. And so as a result, like West Nile virus is more clustered or more prevalent in densely populated parts, in more urban and suburban parts of the state. There are other mosquitoes that are found in more rural areas. So for wet triple E virus, the mosquito that transmits triple E virus is found in swampy habitats, which is found in more rural parts of the state, like in the eastern area along the border of Rhode Island is where triple E virus tends to be a problem.
So currently we are dealing with at least 5 different tick species, each one of them capable of transmitting their own set of diseases. So we are dealing with up to 12 different tick-borne diseases that are currently being circulated in our state. This year we have had higher tick abundance, particularly when it comes to black-legged tick, dog tick, and a number of other tick species that we have. In recent years, we have discovered a number of invasive tick species. 3 of them are the most important. They are Lone Star tick, they are Gulf Coast tick, and they are longhorn ticks. With regards to Lone Star tick is becoming a major problem, particularly in Fairfield and New Haven counties. We are having higher population of this tick species. It is aggressive tick, and it literally chases you, and it is involved in transmission of a number of diseases, including ehrlichiosis and a few other viral pathogens. This tick is also associated with a condition called alpha-gal syndrome or red meat allergy. And we suspect that a lot of people in Connecticut, they might have already developed this red meat allergy. The good news is that we have recently started a research project in collaboration with Yale University, Yale Medical School, and we will soon roll out a project to determine what is the prevalence of alpha-gal syndrome or red meat allergy in our state. As we were investigating these invasive tick species, we came across and identified a pathogen from the invasive longhorn tick. That it is important to note that even though the invasive longhorn tick in its native range, meaning East Asia, and invasive range Australia and New Zealand, is involved in transmission of up to 30 different viral, bacterial, and protozoan diseases. So far here in the United States, we don't have any evidence of human pathogens that this tick might carry and transmit. However, this year we identified a pathogen that is inherently or originally transmitted by Lone Star tick. That is called a pathogen responsible for Ehrlichia. For the first time, we identified that pathogen in longhorn tick. And the reason that this finding is important because both longhorn tick and Lone Star tick, they reside side by side. In a scientific term, they are sympatric. as a result, remarkably and surprisingly, they are exchanging pathogens because these tick species, they feed on white-tailed deer, and because white-tailed deer is a reservoir host, meaning that it maintains and amplifies the pathogen responsible for ehrlichiosis. Therefore, whatever Lone Star tick is capable of transmitting, the longhorn tick is also picking up and transmitting. It is important because longhorn tick is a formidable tick species. It has high ability to reproduce. It is parthenogenetic, meaning it reproduces without reliance on male. In fact, this tick doesn't even have a male partner here in the United States, and it has up to 50 hosts from which this tick takes blood milk, including humans. It also takes partial blood milk that increases this tick ability to transmit the disease agent if it becomes infected during the first partial blood milk. All these things makes us very concerned that if this tick continues carrying this disease agent, we are going to have a major problem, because ehrlichiosis, as some of our state residents remember, it used to be a major problem in our state, and it is a serious disease. And if it is not treated, it could be fatal. Because of all these things, we have every reason to be concerned about this tick carrying this pathogen. Furthermore, we have also isolated in the past couple of years disease agents responsible for Lyme disease, responsible for anaplasmosis and babesiosis from this tick species. Even though for those species, those pathogens, we do not know whether this tick will be competent or capable of transmitting these pathogens, but we now know that at least for ehrlichiosis, it is competent and it is capable of transmitting. It is the matter of time, and combined with high population of this tick species, I would like to emphasize that in some areas that we have this tick species, it takes 10 minutes to walk into those infested areas. When you walk out, you can literally collect hundreds of ticks on yourself. Apart from their high number that causes blood loss and so on for us as humans and for our pet animals. And if they continue carrying this pathogen and perhaps other pathogens, then we are going to be in a difficult position. In the past 5-6 years, we have identified up to 20 invasive tick species that were brought into Connecticut or into the United States through Connecticut by human travelers, unintentionally. Human travelers visited various parts of the world, South America, Central America, Africa, Europe, and you name it. We recently highlighted 7 of those recent introductions to the United States or to Connecticut. And we found out that all those 7 species are capable of transmitting human pathogens of concern, and all these all these species are related somewhat to our existing tick species, including black-legged tick, Lone Star tick, and other tick species. And this is a major concern. Not only these introductions puts our Connecticut residents in difficult position because they are exposed to these ticks that could be potentially infected and they could get sick. There is also possibility that some of these ticks, if they arrive alive here and if they are able to get into the field, there is a good possibility that they might establish population and create a situation similar to longhorn tick, because we believe that we can no longer handle having another or more than one invasive ticks to be coming here. Already the region and our state is plagued with native and invasive tick species and pathogens they transmit.
Most of the experience that I've done with research has been on monarch butterfly conservation. And the work that I'm presenting on at Plant Science Day today is understanding how we should be maintaining habitat for monarch butterfly production in Connecticut. Milkweed is the obligate host plant for monarch butterflies and caterpillars. That means that they need it. So the butterflies will only lay eggs on milkweed, and the caterpillars can only feed on milkweed. The butterflies can feed on lots of other flowers, but in order to have the life cycle continue, we need to have milkweed. Monarchs kind of serve more as an umbrella species or a canary in the coal mine, for a lack of better terms. So monarch butterflies, they are pollinators. They're not as efficient as some of our bee pollinators that we have and other friends like that. But people like them and they have a great experience with them. And if some of our common species are in decline, then there's a lot of species we're not looking at that are probably worse for wear than the monarchs are. So whenever I talk about establishing habitat for monarchs, I always say they need milkweed, but they also like all these other flowers. So we'll establish milkweed and other flowers so then we can be supporting all of our pollinator communities rather than just focusing on the monarchs. So they're the panda bear of the insect world. One of the things for milkweed specifically with monarchs is that a lot of times the common milkweed, the one that has the pink flowers on it that spreads through rhizomes that lots of people don't necessarily like because it will take over gardens, by the end of the season it's flopping over and looking ugly. And then I get lots of calls of, well, when can I get rid of this milkweed? It's bothering me. And the answer is that we can clip it earlier. And then it will be easier to maintain later in the season, but also it's better for the monarchs because monarchs prefer to lay eggs and feed on young vegetative stage milkweed rather than the flowers or when it gets into pods. So if we cut down earlier, then it will get to not be so flopping over in the September time, and then it'll actually produce more monarchs. So oftentimes we'll talk about helping pollinators, and the conversation is help your pollinators plant a garden. One of the things with bumblebees is that the garden, the flowers, will help them over the summer, but in the winter, the queens are overwintering somewhere. They have a hibernaculum somewhere in the ground, usually in like leaf litter or something like that. And then in the spring, they need to establish nests, and those are also underground. So if the hibernaculum habitat type and the nesting habitat type are limiting factors, then no matter how many flowers we put out there, we'll still not be able to support these bees. So some of the work we're doing is trying to find where nests are so that we could identify what the characteristics are about these nests so that then we could provide it to landowners to help have people put that type of substrate out in their backyard, do whatever the things are that the bees like, and then hopefully we'll be able to increase those populations too.
This is our 115th year in a row putting on this Plant Science Day and explaining our science to the public. This started in 1910 when a bunch of people came from the station on a trolley from New Haven to Hamden, and the scientists just stood in the plots and explained their work to them. And that was really unusual, revolutionary at the time, to actually see farmers to see what we're doing in the field and how we're improving crop growth. And we have about over 1,000 attendees a year now. We have people from all ages, men and women, and lots of young adults and college students from schools all over the country helping us present today and attending. It's just wonderful to see. We started hiring a few women scientists around 1904, the early 20th century, but just in the last few years we've gone from 20% to 50% women scientists. We're an important part of the community, and we all work as a team together, so it's great to see our scientists of all different ages, different fields, and different races and genders work together. We serve a very diverse state, and it's important for people to see themselves and the scientists that are serving them. We can serve them in a lot of different languages, where scientists come from 17 countries all over the world, and I think it is important to have that representation. In addition to our 100 full-time scientific and administrative staff, we have almost 100 seasonal research assistants every year. And this is a great opportunity for college students from around the state and around the region to participate in research, get their first foot in the door of what research science is really like. So for our Plant Health Fellows program supported by the USDA, this year we have students presenting their own field research from 8 different universities across the United States, particularly in the Northeast, and none of them have ever participated in paid research opportunity before. And we've seen these students over the last 8 or 9 years join graduate school, join industry, join government research positions. It really helps a lot of them clarify what they want to do with their lives. So I think we're really unique for our small size, and that lends to a collegiality. We can work with scientists from all different fields and get ideas from them. So only having 30 research programs, we really work together as one team, and I think that's what makes us special.
That's all from this edition of Coast and Country from this year's Plant Science Day 2025 and the station's 150th birthday. For more information about the work and research of the Connecticut Agricultural Experiment Station, you can visit their website at ct.gov/CAES. Thanks for watching. We'll catch you again next time.
This goes back to the 1860s and a Yale University chemistry professor by the name of Samuel Johnson. He visited Europe, and Europe had an agricultural experiment station system, which was basically technically trained scientists whose job it was to work directly with farmers, with growers. I started in 1997, and really the largest change is how interdisciplinary science has become in general. But also specifically here at the Agricultural Experiment Station. There's not a single scientist here on staff who doesn't work with other scientists here. And the other thing is kind of our portfolio of domestic and international collaborations, as again, we've tried to do science that's impactful both to the citizens of the state, but in society as a whole.
Women have always been part of the station since its inception. We had technicians doing all of the scientific experiments from the early 1900s. But today, women make up half of the scientists running research programs and about 60% of the technical staff. And so we are doing everything that keeps the station running, from mentoring to running regulatory research programs in public service, doing outreach.
As an agricultural entomologist, I conduct research with pests and beneficial insects in agriculture systems in Connecticut. The work that I do specifically is related to habitat suitability and how insects move across the landscape. Working at a state-level government research facility enables me to design, develop, and implement research that is responsive to state needs quickly.
I think CAAS is important because our role is to do science, and science is about truth. It's about finding solutions to problems that are impacting the citizens of our state and the citizens of the globe.